Today, I'm going to walk you through my project on making a more effective coronavirus vaccine. According to the World Health Organization, there have been over 200,000 coronavirus-related deaths as of May 4th. So clearly, this is a huge pandemic. That's why scientists are trying to find a cure in the form of a vaccine. While a safe, effective vaccine takes anywhere from 12 to 15 years to create, the U.S. government is taking an optimistic stance at 1 to 2 years, according to a Johns Hopkins professor. In the meantime, social distancing is attempting to flatten the curve. In this race for a vaccine, researchers are going away from the traditional vaccine, which gives the recipient a whole pathogen minus the pathogenic portions, and are going towards nucleic acid and peptide-based vaccines, as seen here. I decided to focus on peptide-based vaccines because they skip the sometimes lengthy transcription and translation processes and are therefore faster. This diagram from the Royal Society of Chemistry shows the overall process for creating a peptide-based vaccine. First, I identified the proteins on coronavirus, which I'll explain in depth later. Then, I used the Immune Epitope Database, a free government-funded resource, to identify the epitopes for a potential vaccine. Epitopes are small regions that are capable of driving the T-cell response. What most scientists are doing right now is blocking binding glycoproteins on the virus surface from binding to the cell, as they are directly involved in the infection process. And, as I mentioned earlier, the Immune Epitope Database is currently the best free resource for getting accurate results. Before we dive into the methods, I'll give you a brief explanation of the part of the immune system that I'm concerned with. MHC class I is a protein complex present on all cells that presents epitopes from pathogenic proteins to T cells and generates a memory for the virus, which is the basis of a vaccine. The MHC is highly variable in the population, depending on which HLA alleles a person has. Different HLA alleles code for different MHC proteins, which affect whether a pathogenic protein can be presented to T cells or cytotoxic or helper. That's why it's important to use IEDB to get epitopes that will bind to most or all MHC complexes. To get this high binding coverage across the population without testing every single allele in the population, I used supertypes that were based on the 3D structure of each MHC complex to get epitopes that would bind to more of the population. However, as I reviewed the literature, I wanted to see if there was a better way to design vaccines. The target proteins that researchers at IEDB and other institutions decided to target were almost all binding proteins. Yet, according to Johns Hopkins University, these had the highest mutation rates within the virus. As coronavirus threatens to reappear in the fall, and especially during the colder months, it's important to craft a vaccine that will last in the long term. By using non-structural proteins, I feel that a more efficacious vaccine can be created that is less prone to mutation. Moreover, as seen in the diagram, NSPs make up about 2/3 of the coronavirus genome under ORF1AB. Open Reading Frame 1AB. So, can non-structural proteins be better candidates for vaccine design? If they have enough immunogenic potential in the form of epitopes, NSPs could be better targets for a long-term vaccine due to lower mutation rates. To determine this, I used the epitope prediction algorithms on IEDB, which detect the most immunogenic regions of a viral protein. Okay, materials and methods. For this project, all that I needed was a computer with the applications IEDB and NCBI's protein database. Now the procedure. For methods, the IEDB binding predictions tool contains 4 steps that are required in order to submit. The first step is to get the protein sequence from NCBI and paste it into the query box. This is what the epitopes will be chosen from. Next, I decided to use the IEDB recommended 2.22 method, as it includes all of my alleles and uses 2 of the best epitope prediction algorithms. Consensus combines many algorithms, into one score. An net MHC-PAN is a machine learning algorithm that can predict binding percentiles based on an epitope's affinity for the MHC binding pocket. All of this information is available on the IEDB website. The next step was to input my alleles. These are the same supertypes I mentioned earlier, and the epitope length I chose was 9 because the MHC binding pocket takes 8 to 11 amino acid regions, plus short flanking regions. Finally, I edited the format of my output to make it easier for me to identify promiscuous epitopes. Promiscuous means covering lots of the population through the supertype alleles. I applied this process to non-structural proteins from the coronavirus, and this is an example of the results I got in NSP3, which was the longest protein. This is output from IEDB, showing from left to right the allele, sequence number, start and end of the epitope, peptide sequence, and percentile rank. After that, there are the individual scores from each algorithm. However, those results don't tell you anything in terms of what the best epitopes for a vaccine are, unless you have a good data mining algorithm. To find my needles in a haystack, so to speak, I used a data analysis tool written in Python. This code, written by high school senior Victor Fuentes, shows how improvements could be made to IEDB software by automating the search for promiscuous epitopes and isolating the best epitopes. The final results are these 4 epitopes. Almost all had 100% promiscuity among HLA alleles, making them ideal epitopes for a vaccine. These epitopes would most likely be both processed and attached to an MHC complex in the cell in a real-life situation. Moreover, you may have noticed that these epitopes are length 12 and 13 rather than 9. To get 100% and 87.5% promiscuity, I often had to extend 3 amino acids beyond the selected region to incorporate a binding region for HLA allele B0702. 2 assumptions I made in this project were that IEDB's algorithm was in fact accurate and confirmed by lab tests, and that the epitopes will drive a response in killer T cells in the recipient's body itself. Finally, I found that non-structural proteins are better targets for vaccine design. Advantages of targeting epitopes in these proteins include higher conservancy among strains and sufficient immunogenicity, meaning scientists don't have to reinvent the wheel every year. Moreover, I identified a gap in the IEDB software that could greatly improve user accessibility. Finally, the entirety of this project shows that peptide-based vaccines are more efficient and specific in both design and delivery, and can be rapidly implemented. Even though the peptides I identified were confirmed to drive a T-cell response, there is still work to be done in terms of testing the immunogenicity of the epitopes in the lab by challenging them against human immune cells of different genotypes. In addition, race and pre-exposure could be examined as factors for binding affinity, and the epitope's potential for a diagnostic could be examined. These are the various sources I used to create my presentation and complete my research project. Thanks for watching.